Okay, thank you very much, ma'am. So let's now proceed to the question and answer part of this virtual seminar. So for your comments, you can put your comments at the FB Live chatting page AFRB or NVSU AFRB. So meron pa tayong question muna kay Sir Ruchi.
Yes.
So Sir Ruchi,
Okay. Uh, First, let me congratulate our invited speaker for a very, very good presentation that she had with us for today. Truly, we were amazed with the different techniques shared by Miss Adele from their laboratory. So thank you very much. Uh, in regards to the COS and the CTS, so I am just curious on how you calcified or how is how did you prepare the calcined oyster shell? Is it being calcified, na remove, or ano yung nangyari doon? In the
For the COS, we heat that one. The oyster shell has been heated at high temperature. Yeah, to get this material containing the singlet oxygen.
so it underwent pyrolysis.
Yes. Mm-mm.
Okay. So maybe for the sake, because we were so amazed about the result of the calcined oyster shell and the chitosan.
Mhm.
You see, from Klebsiella pneumoniae at saka doon sa ating isa pang organism, 30 minutes, 2.5 hours, we got 99.999% of disinfection rate.
Yes.
So, in your presentation, you have showed the you have shown the images of the scanning electron microscope and the TEM. Siguro paki-explain lang natin sa mga bata, ano ba yung difference? How do we show the disinfect in what way na naipapakita natin yung mechanism of action of the COS and the CTS doon sa Klebsiella pneumoniae at saka doon sa A. niger via SEM and TEM. Ano ba yung pagkakaiba? Is
Between the T,
ours.
is it between the TEM and SEM, Saruch?
Ano ba yung meron na characteristic ng SEM at saka nung TEM para malaman lang ng mga
Yes.
estudyante natin.
Yes, that's a very good question. I mean, okay, so this is the highlight why we compare the TEM and ECM. For this figure, this is the change in morphology and the ultrastructure of the K. pneumoniae and the A. niger. As we can see, using the SEM, we cannot see the structure inside the cell if we just use the SEM. For the SEM, we will just see the surface morphology, but we will not know what happened inside the cell if we will use the SEM. Okay? So if we use this TEM instrument, we will know what happened, the cells, the structure of the cell inside. Okay, so that's the highlight for this, the comparison of these two instruments between the SEM and TEM. For the SEM, again, we will just only see the surface morphology of the cell or any material, while for the TEM, we will see the cell structure, okay, the inside structure of the cell. So that's the comparison of that two instrument. It's very clear here, right? That for the A Niger, we can just see the structure and we do not know, we cannot assure if the cell has been damaged inside. Because, yeah, and for the TEM, it is very clear here that using the TEM, we can see that the cell has been damaged, right? So that's the difference between the SEM and TEM.
Thank
I
you
hope
very
I
much.
answered the question correctly.
Yes, thank you very
Okay.
much, Miss Mel. Kasi syempre, we all know wala tayong, meron in our university, I'm glad to inform everyone that we already have the scanning electron microscope. But the
Wow, that's great.
TEM, wala pa. Actually, nakita ko lang din ang TEM sa Taiwan, in your laboratory.
Oh,
Yeah.
okay.
So, maybe yung ating mga BS Bio students also are interested about this biodegradable mulch. Kasi baka pwede nilang gawin later on in their thesis regarding the biodegradable mulch. Uh, can you please explain to us kung paano ginawa uh,
Biodegradable mulch.
yung biodegradable mulch? Yung,
Okay. Let me show
kasi
you this.
pwedeng-pwede nilang gawin na thesis yun, right?
Yes,
In the
can.
later on in their thesis.
So I think this will answer the question.
Yeah.
Okay, so how did we synthesize this biodegradable mulch? Okay, so as I have mentioned, we use three different agricultural waste, right? From the agricultural waste, the shrimp waste rather, and the chicken feeder. Okay, we all know that, especially for the chicken feeder, because in the Philippines we are consuming chicken than pork because chicken is cheaper than pork, right? So probably there are many chicken feeder waste. Okay, so what we do here for the biochar, yeah, we synthesize first the biochar under the paralysis condition, and for the biofilms, it undergoes. Let me. It's here. No. For the biofilms, so actually, in this for the chicken feeder, our lab doesn't made this one. This is was from another lab. So, but it undergoes a process also. But now, right now, I cannot provide the answer just because um, this chicken feeder was synthesized by the other lab.
Yeah, I
Yeah.
understand, Mel.
Mm-mm.
Isabihin lang natin sa ating mga viewers kasi how our each university in National Chongqing University, actually not only in National Chongqing University works. So, for the sake of in each laboratory, we usually collaborate with other laboratories.
Department.
Okay, so
Yes.
hindi
So,
lang iisang laboratory ang
mhm.
nagpro-produce ng isang study. So, it's a
Yes.
collaboration and
Collaboration.
sharing of resources. Okay,
Yes.
para alam din ng ating mga viewers. So,
Mm-mm.
yung portion ninyo is you made the biochar, right?
Yes.
And in the
Yes.
synthesis portion, the other laboratory did that for you. And then, pag natapos, bumalik sa inyo for the investigation. Tama?
Yes.
Okay,
Okay.
so
So
ganun
this
yung
is
nangyayari
the.
yung
Mm-mm.
collaboration and research doon sa Taiwan. So ganun po yung research environment doon. Unlike
Yes.
kasi
And different expertise.
information for the sake of everybody na hindi po kasi tulad dito sa atin sa Philippines na for example, you are from the College of Agriculture. So your thesis is just limited with
Within the
what
college.
is being served in
Mm.
your department. For example, you are
Mm.
in soil science ka lang. If
Mm.
you are in pathology ka lang. So kung BS biology ka, hindi ibig sabihin na yung pagiging BS bio mo, contain ka lang sa mga common or typical BS bio na courses. And that's the reason why we try to invite different researchers across the fields kasi nga yung bio course natin across all fields pinapasukan niya lahat. So, ganun yan.
Mm-mm.
Okay, thank you very much, Miss Mel.
Okay, so I would go back to the how did the biodegradable mulch, Saruch? Okay, so we do the extrusion blends. So we blend all the materials, and to make the films, we use the hot pressing here using this instrument to make that biodegradable mulch film. Okay, and then yeah, this is the biochar polymer composite. This is the product after the hot pressing. Yeah. So first, again, we combined all the ingredients. It's like an ingredients, like when you're cooking. 'Cause- So the biochar, the chitin, and the chicken feeder were combined. Okay, and then yeah, it undergoes hot pressing using this instrument to get this product. So that is how we synthesize the biodegradable mulch.
So thank you for a very good presentation, Ma'am Mel. So the students would like to express their appreciation and commendation on your very comprehensive presentations. So these students will be conducting their research thesis or later, so they were very eager to ask this question. So in relation to your presentation or your expertise, can you tell them a possible research to explore, especially in the Philippine setting? Thank you.
Um, for my presentation, I think they can use the idea for the disinfection mechanism since most of the students are the degree is Bachelor of Science in Biology, right? So I think they can apply for that. And for my new research, I think they can also my research in the soil biological properties. Okay, let me give you maybe I can share to you my current research. Why it's not moving? Wait a moment. Okay, by the way, my current research is about the soil quality index in agricultural soil. So this research would involve the soil physical, chemical, and biological properties. And I think the students can do they can apply for that soil biological properties because I will be analyzing the enzyme activity in the soil, and they can apply that research. For example, my research in the Philippine setting because I think we have the instruments. The instruments are available in our country. Yeah, and hopefully we can buy more instrument so that we can answer all the like question, maybe the mechanisms because mechanisms is important. No, why this one decrease? Why this one is increased? And we cannot just say that one increase decrease. We must give a mechanism why this thing happened. Yeah. So did I answer the question? Yeah. And I am very hopeful that in the future or maybe next year, that our technology would also improve, more improve. Like here in Taiwan, Taiwan has the national synchrotron, synchrotron NSRRC, where we used to analyze our sample using the 3D tomography.
Thank you for that wonderful answer, Ma'am Mel. But again, we have another question coming from our department chair, Sir Dan.
Hello, sir.
Um, yes, madam. Good morning. Thank you very much for your brilliant insights and ideas. I would just like to clarify if your research is towards the development of an antiseptic or a disinfectant.
Disinfectant, sir.
Disinfectant. I'm just curious if the process of producing that disinfectant would be cost effective since basing on what you have presented, I think the process of producing it would would be costly. I just don't know.
Um, I don't think this is costly, sir. The for example the CUS derived from the shrimp waste. Um, in our lab, we just clean that one. So it's the clean and then just hit the oyster shell, and then we will get the material. And we have this instrument. We have this instrument that able to to make it smaller particles. I would agree. It is costly. It's because of the instrument, just the instrument. Like for example, the how to make that oyster shell become small, become nanomaterial. But I think if you just invest the instrument because it is beneficial, it would benefit. You don't for the disinfection. You don't need to use chemical. Just use utilize the waste from the fishery.
So I'm praying, Madam, that this will be a reality in the future. So I'm praying that God will bless you in your research. Thank you.
And so actually, we joined several symposiums or conference, and we already provide to the public. We already distribute our product, the disinfectant derived from COS. Yeah. We already share to the community or to the people during the conference or meeting.
Thank you very much ma'am, for that update.
Mm-mm. Mm-mm.
Thank you, po.
You're welcome, sir.
We have another question coming from one of our faculty members. Sir Richie, you are recognized.
I'm just curious about the COS and the CTS, Miss Mel.
Yes.
How did you expose or how did you utilize the COS and the CTS as disinfectant? Is it in a liquid
Yeah,
media
that's
and
a
then
very good question. It's a
just pour
liquid,
into the bacterial culture?
liquid, liquid syruch, liquid form.
So what is your solvent? Just pure water?
Yes, the ionized water. We have this the solid. I mean from the C2S, and we just dilute that one into the ionized water. With the I forgot the ratio. We have the ratio for that between the solvent and the solute.
Yeah, and of course from there we can also maybe later we can determine the dosage and of course the minimal bacterial MIC and minimal
MIC.
inhibitory
Yeah. Inhibitory.
concentration.
Yeah. Actually, we have results for that, the MIC, but I just did not show because in this presentation I focus on the mechanism. We have a result for that, the MIC, microbial inhibitory. Is that concentration? MIC, if I'm not wrong. Yeah,
Yeah.
we have a result for that. At this part, our research is more focused on the disinfection mechanism because we have to prove that our product is really effective for the disinfection of the specific microorganism. Thank you, sir.
Question po.
Yes,
Yes, sir.
Ma'am
Question
Judith, you
po.
are recognized.
There. Thank you. First and foremost, congratulations, Miss Mel, for a very informative and very timely research. I would just like to ask, I don't know if I missed it because I was kind of busy a while back. Was there a preliminary study or a related study as to the possible active components of our chitosans and the oyster shell? What's that again? I'm so sorry.
Oyster shell
Do we have?
and yes,
Yes.
yes, we have it, ma'am, in our lab. We have a complete
Mm,
detailed information. We have a complete results about the
mm,
properties or the as what I have mentioned from Sir Roach, the MIC. We have all
mm.
that preliminary results in our lab before we proceed to this mechanism.
Uh, Ma'am, with that, what is the like the major or the how do you call this? Yung pinakmataas yung percentage na composition. Ano yung
It
pinaka-active ingredient kumbaga?
for the oyster shell, it is the singlet. I don't know if I pronounced it well. The
Singlet oxygen.
singlet
Uh-huh.
oxygen for the that is for the oyster shell, the COS. While
Mm,
for the CTS or the chitosan derived material,
chitosans.
it is the amino groups. The amino groups. The
Mm,
let me show you.
yeah, yeah.
Yeah, it's amino groups.
Okay. Kasi mostly kasi itong chitosans natin and the oyster shell. I think the oyster shell is more on calcium.
Calcium, yes.
Meron din siyang calcium, 'di ba? Tapos si chitosan naman natin is complex proteins.
Yes, yes.
Yes, complex proteins. Kasi it covers the backbone. So since you have already like related studies, so were you able or is it a recommendation that we be able to define or look for the mechanism of the disinfecting property of these active ingredients, or is there have you tried mixing the two?
Not yet, ma'am, but in the future we are planning to combine these two materials
Yes,
to have
yes.
a more result. But right now we just first study each of this material.
Yeah, so possible future research din po 'yun, ano po?
Yes, yes, ma'am.
Kasi we can compare like do they work better alone or do they
Or
work or do they
combination.
produce they have synergistic effects? Ang tawag ito, for our undergraduate studies, so these are possible research areas that you can actually explore, especially for our med bio students. You can tap or you can check ano yung possible aside from disinfecting properties. Are there other properties that this, let's say, how do you collectively call it, fisheries waste?
Yes, fishery waste.
Oo, kasi hindi naman natin kinakain tong mga to, di ba? Like today, mag-uulam kami ng I don't know how you call it, pero we call it agurong in Ilocano. So, paano natin siya i-dispose as a waste, di ba? Is it biodegradable? Is it non-biodegradable? Kasi if you notice, matagal siyang mag-decompose.
Yes.
So, yun. So, for sa ating mga students, so these are possible research areas that you can tap. And we are very thankful for our speakers for sharing their time, their expertise, and for accommodating us in this research. I mean, and of course, kudos to our proponents. Siyempre, yung ating department chairs. Sir, thank you, ma'am.
You're welcome. It is my pleasure.
Okay, so we can entertain more questions coming from our audience. Yes, Sir Dan, you are recognized.
Yes, ma'am. Pahabol po.
Yes,
I
sir.
just forgot to ask you, for every kilogram or for every weight of your waste material, how much of your disinfectant would result? What's the yield?
Actually, we did that, sir. We have the result for that, but I do not have here. But maybe I can answer that by email. We have
Okay.
yeah, yes, sir. Sorry for that. But we analyzed that one, like for example, how many kilograms of waste we got from the industry company. We weighed that one, and then how many grams is the product or the disinfection rate ratio, whatever.
Okay.
Yeah.
Thank you, ma'am.
Sorry, sir, I
And
cannot
one
answer right now.
Yes, ma'am. And one last thought. Since our context in Viscayas, we are an agricultural province.
Yes, sir.
So it's more on waste from the rice field uh, and or any agricultural waste product. What particular waste product do you think would be a good material, which could also be subjected to the processes and experiments that you have undertaken?
Okay. So based on our results, sir, in comparison with the five feedstocks, the rice straw, rice husk that we use in the lab, rice straw, rice husk, corn waste, and the sugarcane bagasse, and pineapple leaves, between that five feedstock, our results show that the pineapple leaves shows a good properties which the soil can or the soil or the plant can benefit because of its unique properties. The pineapple leaves shows a very good exchangeable basis and high pH. Also, the functional groups. Yeah.
Okay, so that's good. For where you have talked about pineapple leaves, right?
Yes. Yes, sir.
Yeah. So here in Vizcaya, we have an area planted with pineapple. So that will give our students an idea on what
Yes,
to
sir.
work on in the future.
Mm-mm.
Well, thank you very much.
Yeah. And I would like to add, sir, because actually there are many research also the rice straw can be used, but we use the pineapple leaves. Yeah, pineapple leaves is good, better, but we can you can also uh, use the rice straw. It's also good. Yeah,
Thank you very much, ma'am.
you're welcome, sir.
So at this juncture, we shall proceed with the awarding of a certificate of appreciation. But before we do that, may we hear some announcement from Sir Tyron?
Before we proceed to the awarding of certificates, let me just remind you na pwede kayong mag-evaluate sa ating seminar series. O yung link po ng ating evaluation form ay posted sa EFRB and VSU AFRB page and nasa comment section na rin po atin. Okay? So let's proceed to the awarding of the certificate. So we have here Dan with the Department of Social Sciences. Okay? He will present the certificate of appreciation to the two speakers. So let me just read the contents of the certificate. So, Nueva Vizcaya State University, College of Arts and Sciences, here at Bayambong, Nueva Vizcaya, Philippines. So, this certificate of appreciation is awarded to Sir Brian James, Sir Ruchi Brian James Lagitnay and Ma'am Mel Adele Okba for sharing his and her expertise as resource speakers during the virtual seminar series in biology, biochemistry, and molecular biology held on December 13 and 17, 2021 at Nueva Vizcaya State University, Bayambong, Nueva Vizcaya. Given this 17th day of December 2021 at ITE Lab 301 and VSU Bayambong, Nueva Vizcaya. So signed by the Department Chair of the Natural Sciences, Sir Daniel Marvin Ramel, the College Dean, Dr. Philemon A. Pamitan, Campus Administrator, Ma'am Cristina R. Salvosa, Vice President Affairs, Ma'am Jocelyn P. Cabrera, and of course, the officer in charge of the Office of the University of the President, Honorable Ruth R. Padilla.
Thank you.
The same certificate shall be awarded to Madam Mel Adele Okba.
On behalf of Ismail, ako munang tatanggap.
Okay. Thank you, Sir Rich. Thank you, sir. Thank you.
To formally end our two-day seminar series, may we hear a closing remark from our department chair. Sir Dan.
So it's I think a long week for us, though this is just a 2 day seminar. So may I just reiterate again my profound gratitude to our esteemed speakers for their time, for their sacrifices. Okay, and I believe the information, okay, that you have imparted to us would greatly help our students in their future undertaking or research. So I pray that God will bless our speakers to become blessings always to the community and to others. For our BS Bio students, I believe a lot of you were overwhelmed by the level of research presentations that you have heard or witnessed. But I believe also that this will also open opportunities for you to be able to interact, collaborate with these speakers so that you can better understand specific areas of research that you wanted to pursue in the future. So I hope this will not end, okay, but will drive you to pursue, okay, good research once you take your thesis writing by next semester. So I will not take you for long. That would be all. God bless sa inyong lahat. Merry Christmas and a Happy New Year.
Thank you.
Before we finally end this Google meeting, may we request all the participants to turn on their camera for a screenshot or a picture
Yes.
taking with our speaker. Thank you so much.
Hello. You're welcome, sir. Okay.
Thank
Okay.
you,
Thank you.
Miss.
Bye bye. God bless. Stay
Bye-bye.
safe. Oh my God.